Good afternoon. Let's go ahead and get started. It's really a pleasure again to welcome you here to the Dean's Distinguished Lecture. Of course, we're just so delighted to have Dr. Cepko here today, who will give her presentation, Gene-Agnostic Gene Therapy to Prolong Vision. Of course, as an ophthalmologist and a neuroscientist, this is an area that's particularly of interest to me. And again, we're so glad that you're here with us. Before we go any further, I would like to acknowledge our Emeriti faculty who joined us earlier for a lunch. So please maybe stand, let everyone see you if you don't mind. Um, and again, we're so happy that they're here with us. They've of course over time have contributed so much to our college about who we are and what we do, and again are still very engaged in our College of Medicine. We launched this lecture series in fall of 2022, post-pandemic, to highlight research topics which integrate efforts by multidisciplinary teams. And we know that science in many ways is a team sport, and that we come together around omics, high-end imaging, artificial intelligence, molecular biology, population health, all sorts of things that come together now to make great, impactful science happen. And the point of this lecture series, of course, is to bring eminent scientists, world-class scientists from around the world to tell us about their journey and their work, also to bring us together in ways that we might not have done so much during the pandemic. And I'm so delighted that you all stay for the lecture, but also come to our reception that follows. I will say it's also very important to understand today we have one of the world's very best neuroscientists who's made fundamental discoveries in many things. And I know now there's a translational application. We'll hear more about that. And I just like to note that translational science can't be replaced by not such good science. There has to be at the base of this, a core of this is outstanding fundamental science that then can be translated into future cures, therapies, diagnostics, etc. So with that, I'd like to introduce for the more formal introduction to Dean Prabhakar, our Senior Associate Dean for Research, who is instrumental in bringing Dr. Cepko here today.
Thank you, Dean Rosenblatt. So it's an absolute pleasure for me to introduce our speaker, Connie Cepko. It's a very strange incident happened about nearly 40 years ago. So I was at NIH and I was asked to be on a site visit team for a program project proposal from Dr. Bernard Fields. Many of you may know him. He's the guy who edits this thick virology textbook. And Bernie is a phenomenal researcher, an incredibly wonderful human being, and he was the PI of this PPG. So I happened to be on the site visit team with Stanley Prusner. Some of you may have heard of him. He went on to win Nobel Prize for discovering prions. And he was the chairman of the committee, and then we go there, and all the PIs, make their presentations. And there was Bernie Fields, and there was Howard Weiner, and then comes this very young lady called Connie Cepko, and she makes her presentation. And late afternoon, and we all go back to our hotel, and we're all sitting around the table drinking some beer. And one topic on everybody's mind was this amazing young person who had blown all of us away by her presentation. That young person happens to be Connie Cepko. So we all concluded at the end of our dinner that she's going to be a superstar. So it speaks to her own abilities to become a superstar. And maybe slightly we'll take some credit that we had good judgment to speculate that. So with that personal anecdote, let me move on to tell you a little bit about Dr. Cepko. She's a Bullard Professor of Genetics and Neuroscience at Harvard Medical School, and she's an investigator at the Harvard Hughes Medical Institute. She received her PhD degree from MIT, working with Phil Sharp, and remained at MIT as a postdoctoral fellow in the laboratory of Richard Mulligan. I mean, my God. And where she was involved in the development of retroviral mediated gene transduction into the nervous system for lineage analysis and for studies of gene function in vivo. a little bit of a slight anecdote here is that by that time Phil Sharp wanted to actually look at splicing, and Connie was not interested in that. So a lot of the work that she ended up doing there was literally largely her own, and then that was extended in Richard Mulligan's lab. And so her current research is focused on the development and diseases of the retina, addressing questions regarding the mechanism of self-weight determination through the analysis of behavior of progenitor and stem cells, and developing gene-agnostic gene therapy to prolong vision. I mean, that's a pretty loaded sentence. There are about 4 areas of serious investigation there. She's a member of American Academy of Arts and Sciences and the National Academy of Sciences. She has received innumerable awards for both her research and mentoring. She has launched and directed 2 PhD graduate programs and is currently serving as the co-director of Leader Human Biology and Translational Medicine program. Without further ado, Connie, we welcome you here.
Okay, thank you very much for that kind introduction. I didn't know that story. I appreciate that. And thank you all for coming. I've had a great day today, meeting with people, and last night as well. So thanks for being such an interesting group. I had all kinds of great conversations today. And as you heard, I'm going to tell you about our efforts to create some gene therapies that hopefully will prolong vision across many different disease gene families. And I like to start with this slide because it shows sort of the star of the show, the cell type that we're trying to preserve. This pointer's acting funny. Okay, can you see my pointer? Yes, you can. Okay, this actually is a cone photoreceptor. These are the cell types that initiate our color and daylight vision. It's reconstructed from a mouse. And this green portion out here is called the outer segment. And this is where the phototransduction machinery, the rhodopsin protein that captures light is all housed. It's a very, very specialized structure, and it's required to have high efficiency capture of light and processing of the signal. Then on this inside portion here, you see this red spot. This is an epitope of phospho-mTOR. So mTOR is a central regulator of metabolism. When it's phosphorylated, all things are good. The cell gets a green light to grow and to metabolize. It's also housing a lot of mitochondria, which are very dense in these photoreceptors. And this purple area is where the nucleus and the surrounding thin shell of cytoplasm resides. So these photoreceptors are these highly specialized cells. They're very sensitive to genetic and other insults. And there's over 300 disease genes that now lead to loss of vision. Most of them are genes that are expressed either specifically or at least in these rod and cone photoreceptors. So cone, again, is the one I showed you for daylight. Rods are the ones that are important for night vision. Now, here's just some of the disease genes. I'd like to introduce the disease that we mostly study. This is called retinitis pigmentosa, and you can see the names of many genes. We're over 100 different genes now. Most of them are expressed only in the rod photoreceptor. So rod photoreceptors are active in dim light conditions. So if rods are not functioning properly, a person has poor or no night vision. What happens after that, however, is what really impacts quality of life, and that is the cones also degenerate. So rods often degenerate due to intrinsic problems. The disease gene is expressed only in rods. But once they pass, the cones, which do not express the disease gene, can also die. And then a person loses their color vision and their daylight vision. And that's the target. That's what we hope we'll be able to address by using these gene-agnostic treatments, because those cones are suffering from bystander effects. Again, we don't have to correct a disease gene within cones. They don't express it. But if we can improve whatever the conditions are that lead to their demise, we hope that we can prolong our most valuable vision, which would be the color and daylight vision. So here's just a picture, a micrograph of some of the cells we're trying to save. Again, these are real micrographs. This is in a human, and these green cells are the cones. The rods, and the blue cells are the cones. You can see their cones from the shape of their outer segment. So again, we want to save these cells, and we're trying to come up with strategies. And for that, we have to understand, why are they dying? So part of my talk will tell you our attempts to understand why are they dying, and then some of our educated guesses based on that to provide gene therapy to combat the problems that are causing their death. So we can think of it in a fairly simple schematic way. So down here is the period of rod dysfunction followed by death. So this pace of rod degeneration varies quite a bit. Even in a family with the same disease gene, an individual might start to lose their color vision, which happens here, at age 20, and a sibling maybe at age 60. So there's this wide range. We don't know why. It could be environmental. It could be modifier genes. But the problem is that these rods die, and then the cones get sick and die. So again, night vision's first, and then the cone vision is lost. Now, again, why is that? So there are many theories in the literature. There's observations that are correlated. We don't always know if they're causal. And here's some of the things that we have thought about, and others as well. One possibility is there's a lack of trophic factors perhaps produced by rods that cones need to survive, and there's some evidence for that. It's possible that toxins are released by dying rods, and those affect the cones. I don't think that one is true. We got 4 mouse models of retinitis pigmentosa, and we charted quantitatively these 2 periods, rod versus cone death. And you can see the cone death starts after almost every rod in the neighborhood is dead. And if toxins were being released, we think that would happen closer to the peak of rod death. So I really don't think toxins are part of it. Oxidative stress has been measured in the animal models as well as in humans. I'll tell you about metabolic problems that we discovered. And there's always inflammation. Every neurodegenerative disease or many diseases are accompanied by inflammation. So these are the things that we think we have to combat to keep the cones alive. So we'd like to be able to introduce some of the genes that we hope will fight these things when the cone death starts. And the reason we want to wait until that period is because, as I told you, there might be someone fully sighted until age 60. So we really have to wait until a lot of rods are dead to feel that the risk is justified to introduce a vector. So the way we started was to get these 4 mouse models, which we got from our colleagues. These are mouse models that have the human disease genes expressed as a transgenic. And we got 4 of these models. And we then looked at the RNA changes across these 4 models, all of which have, again, different disease genes or alleles and different pace of degeneration. And we looked for changes that were in common when the cones started to die. And our logic was that's when cones start to suffer. And again, we were hoping it was disease gene agnostic, that is, common problems across the cones in all 4 models. And indeed, we found a common signature, and it had to do with metabolism, which we hadn't been thinking about at that time. So we started to then look at what's happening to metabolism in the cones. And one of the things we found was that mTOR was not phosphorylated. So I showed you on that very first slide that red spot in the inner segment where the mTOR resides. And mTOR, again, is this central regulator of metabolism. And there are inputs to mTOR's activity. When it's phosphorylated, it's active, and it basically itself is a kinase, and it gives a green light to growth through regulation of these basic processes. When it's not phosphorylated, it could be because there's not enough growth factor signaling, which feeds into it. There may not be enough building blocks to support all this this anabolic activity. ATP and AMP are also important for energy utilization. So when it's not phosphorylated, it's a sign that there is some kind of limitation. We don't always know what. And it can also act as a transcription factor or regulate transcription factors. So HIF1 and the glycolytic pathway goes up, the glucose transporter goes up. Essentially, it seems that the cell is trying to get more energy when mTOR is not phosphorylated. So we looked at this in cones, in these degeneration strains, and in all 4 strains, the first sign of trouble we saw for cones was lack of mTOR phosphorylation. And we also found that these genes, HIF1, GLUT, and glycolytic genes were up. So it looked like there was some initial problem. Again, it could have been receptor signaling, it could have been lack of nutrients. To start to look at this, we thought, well, if it's lack of growth factor signaling, perhaps by injecting the animal models with insulin, we would stimulate phosphorylation, and thereby we would save more cones. And indeed, that's what happened. This was work from Claudio Punzo, my postdoc at the time. So we either took away the pancreas with a drug, STZ, or we injected insulin every day for 3 weeks. And what we saw is shown here as what we call retinal flat mounts. So these are the inside of the eye. We strip out the retina, and we flatten it out by making a few cuts. And here we're looking at a cone marker that's shown in blue. And what you can see in this mutant, which loses rods first and subsequently cones, blue shows us where are the cones that remain at this time in this mutant. And you can see most of the central cones are gone. If we inject insulin, we get more of the central cones to survive, also better survival out in the periphery. And if we take away the pancreas with the STC drug, we get fewer of these cones. So I don't think this is a good strategy for a treatment, but it gave us the idea that maybe we should pursue this line of investigation further, that there might be a problem due to lack of mTOR phosphorylation. And subsequently, Claudio Punzo, who now has his own lab, showed that the problem was intrinsic to the cones. That is, we were injecting insulin systemically. It could have affected the cones in many ways. But in fact, by manipulating gene expression within cones genetically, he showed that this effect was actually in the cones. So then it came up, why is the mTOR not phosphorylated? Now, we didn't know, we still don't know for sure, but we have a model. And what we think is that it's not getting enough glucose. So rods and cones are very dependent upon glucose. We took out some fresh wild type retinas and we cultured them just for a short period of time, right after we explanted, in a medium without glucose or with glucose. And then we assayed for the phosphorylation epitope on mTOR, and you can see it's present when we have glucose, it's absent when we don't have glucose. Now, this again doesn't prove what's happening in vivo, but it made us think, well, maybe the cones aren't getting enough glucose. And again, we knew photoreceptors depended upon glucose. So we started thinking about, well, why would they not be getting enough glucose and what could we do about it? So I want to tell you a little bit about where they get their glucose from and why this might be a cause of their problem. So what's diagrammed here, this big cell is called a retinal pigment epithelium cell, or RPE. This cell is interposed between the photoreceptor outer segments and the vasculature called the choroid here. The choroid gives glucose through GLUT1 transporter to the photoreceptor. So the RPE takes it in and passes it on to through the GLUT1 transporter to rods and cones. There is no other vasculature that supplies nutrients to these photoreceptors. It's all coming from the choroid through the RPE. Now, once the glucose is here, we also know that these photoreceptors carry out what's called aerobic glycolysis. This was actually first noted by Warburg, that in the presence of a lot of oxygen, they still carry out this aerobic glycolysis, and they secrete lactate. So because they're secreting lactate and they're not using it, it can be picked up by these RPE cells. They have their own transporters for lactate on this side, the apical side, as well as the basal side. They can release lactate if they've taken up too much. And presumably, if they don't have enough, they could take some up from the blood. So they're using the lactate then, they convert it to pyruvate, and they run through the TCA cycle, and that's where they get their ATP and their building blocks. Now, what happens then when there is degeneration? Just going to skip ahead to that. So when there's degeneration, their source of lactate goes down. So I told you there's rods and cones, and in the mouse and in us, 95% of our photoreceptors are rods. So they're the major supplier of lactate to these RPE cells. So here's what would happen in a normal healthy retina. Glucose comes into the photoreceptors, lactate goes out, it goes to the RPE. They use it for their metabolism. This allows them to give up the glucose to the photoreceptors. Now, in the disease case, the rods die. And I told you that cones really don't start to die until almost all the rods in their neighborhood are gone. And we think that's what's happening is that because there's now a deficit in lactate production or availability to these RPE cells, they keep the glucose. They don't give it up to the remaining cones. So then the cones, not getting enough lactate, start to degenerate. So basically, it's a lack of supply because the RPE is responding to lack of lactate, which they would use for their own metabolism. Okay, so we thought, okay, if that's the problem, perhaps we can supply more glucose to the cones. How are we going to do that? So we have three different ways to do that. The first one I'll tell you about is a little bit out there. This was an idea that Claudio Punzo had, and that was to give the cones the ability to make their own glucose. To do that, we used AAVs. So most of you probably know that AAV is a vector of choice for delivering genes for therapy as well as for basic science investigations. These are these small DNA viruses, and we can grow them in the lab pretty easily, and we can introduce them by injecting them in vivo into the eye. We usually go to a neonatal animal, and we inject such that we get the viral inoculum to flow right through the area where the photoreceptors and RPE resides, called the subretinal space. And we actually put in a couple at the same time. So the green virus depicts a virus that has a red opsin promoter. It's cone-specific, and it drives expression of a nuclear GFP. Now, that gives us the ability to easily count the cones because we have this very bright nuclear stain of GFP. Then we co-inject 1, 2, or 3, or 4 other viruses that might have therapeutic potential. So they're all going in at the same time. And at this time, with the doses we inject, we can get very high rates of co-infection. We can test that with putting in, for example, a red and a green virus, and we see that all the cones are yellow. So we put these in at the beginning, and part of the reason we also do the beginning, not just because we get really good inoculum flow on the young eye, but some of the genes, some of the models we're studying for retinitis pigmentosa have very rapid degeneration. All the rods are gone by 3 weeks of age, and these AAVs take a few weeks to ramp up their gene expression. So we're trying to get the genes in there before degeneration. We then take the animals, remove the retina or the RPE, we make some cuts to flatten it out for the retina and the RPE, and then we count the number of cones, and we also do a visual acuity test called the optomotor test. So this is a live test where we put the animal in a drum that has simulated circulating stripes, and you can tell what kind of acuity the animal has by changing the width of the stripes. The narrower stripes, the higher the acuity. So we can ask whether we preserved vision. The animal basically tracks with its eyes or its head as the stripes go by if it can detect the stripes. So by counting cones, and we usually do the center because, as I showed you earlier, most of the cones die first in the center and then out to the periphery later. So we count the cones, we do the optomotor test, and we ask if we introduce genes that allow the cones to make their own glucose, do they live longer? So here's the genes we introduce. This is a pathway that might be familiar to most of you. This is glycolysis. It has a number of enzymatic steps. Now, to reverse glycolysis in a process called gluconeogenesis, you need 3 additional enzymes shown here in red. Now, this is normally only carried out by the liver, and we checked, and these enzymes aren't present in the retina. So we put these 3 genes into actually 2 different AAV vectors. We could fit 2 of them into one. And then we injected these mouse models to ask whether the cones would live longer. And indeed, they could. So these are now counts of GFP-positive cones in the center and the periphery of one particular disease strain. So on the y-axis, we have number of cones in that central area. This is in the center of the retina. This is in the periphery. Green is a GFP-only control. We don't have many cones left at this assay point. This is a transcription factor I'm going to tell you about later called NRF2, which fights oxidative damage. This is the survival of cones when we have the gluconeogenesis genes on board. And this is when we put all 4 vectors in, NRF2 plus gluconeogenesis. It's additive. And it's true also in the periphery. So this made us very happy. It seemed like at least it was supporting the hypothesis that the cones had a metabolic shortcoming. We're not sure why still, but we thought, okay, well, at least it's allowing us to address the survival problem. So that was one approach to this. Then we thought maybe if we gave the RPE more lactate, it would release the glucose, which it normally does when it gets a lot of lactate. So to do that, Laurel Chandler, a postdoc in the lab, made a vector that had a high affinity lactate transporter. So I told you then lactate's produced by photoreceptors. It can go into the RPE through a lactate transporter on this surface, on the apical surface called MCT1. It can be released with another one called MCT3. And here's their relative affinities for lactate. They're not particularly high. And we thought, well, maybe we could give the RPE a higher affinity one, which would allow it to take up lactate from the blood. And the one we chose was called MCT2, which you can see has about a 10-fold higher affinity for lactate. So we put that one in with an AAV. This time we put it into the RPE, not into the photoreceptors. And there's a very nice promoter called the best one promoter, RPE specific. It's small, it fits in the vector very nicely, driving the MCT2. And again, as a control, we have GFP only. And so these are some flat mounts with cones being stained in 2 different mouse models. This one's called FEB, it's very rapid. And this is a rhodopsin mutation called P23H. These are both representing human mutations. And on the left is the remaining cones from the control, GFP only. And here with MCT2, you can see we have more cones. And these are the counts where we see there's more cones. And we also looked at vision using this optomotor test. And this is the acuity. It's better if we have the MCT2 on board. And it does, unfortunately, wear off. As the animals age out, we do lose some of the benefits we get at the earlier stages, which seems to be true for all of our treatments as well as everyone else's treatment in the field. So I think it's because we need to do combinatorial gene deliveries to get the best benefit. Okay, so we were wondering, what are we really accomplishing by putting in this MCT2 transporter and these gluconeogenic genes? We tried to do metabolomics by taking out tissue and paying a lot of money, get people to run metabolomics, and we couldn't get reproducible results. And we think it's because As soon as you take the tissue out of the eye, everything changes, and it's hard to dissect at the same rate and get the same kind of populations for the metabolomics. So that turned out to not work out. But meanwhile, people were developing these beautiful sensors for metabolites that you could then introduce into cells, and we did this again with AAV. They're called fluorescent lifetime sensors, or FLIM. And these are usually circularly permuted fluorescent proteins like GFP or T-sapphire. And people have looked far and wide for genes, usually from bacteria or yeast, which will bind a small metabolite like glucose or lactate or ATP. And you can create fusion proteins between the GFP and these binding proteins such that you create this kind of a fusion. And when the fusion protein is not bound to the ligand, again, let's say lactate, it doesn't have a long fluorescent lifetime. That is, the fluor is excited and it very quickly goes down to ground state, or the opposite. Some actually have a longer half-life when the ligand's not bound. So by looking at the fluorescent lifetime, again, more ligand, often more longer fluorescent lifetime, you can ask whether there's more lactate or more glucose in the treated cells. So we did this with a lactate and a glucose sensor, and this is how it looks. So we infected in vivo with the MCT2 vector. And as I showed you, we have longer life for the cones. We have optomotor. And now we want to know, do those cells actually have more lactate? So this is a flat mount. We infect in vivo. We take out the RPE, very quickly put it on, run to the microscope, actually, and then image the sensor. By the way, we introduced the sensor at the same time as the MCT2. So then we add lactate at various levels while we're imaging. This is the control, which has just GFP. This is the MCT2 with GFP and MCT2. And this color indicates the fluorescent lifetime shown here on this chart here. And what we can see, lactate is one where you actually have a shorter half-life with more lactate. The red shows us the half-life, the FLIM half-life, with the MCT2 on board, and blue with just the GFP. And you can see we have better, it indicates that the cells have more lactate if we have MCT2, which is exactly what we were hoping for. Now, I can't still prove that that's why they live longer, but that's the only known function really for MCT2. Well, I should take that back. It has a very good function for taking up lactate. It can take up some other small molecules that have the same sort of number of carbons, and so we're also interested in looking at whether they take up more of those. But for here, this was good. It showed us that, yes, it looks like they have more lactate because we're adding lactate to the media. And we also looked at the glucose sensor called Sweetie. In this case, longer FLIM reflects more glucose intracellularly. And again, here's the control, and here's the MCT2. Red means more glucose intracellularly. Here's the quantification. Now here, we're not sure what's going on. Again, we're putting in a lactate transporter, but the cells have more glucose. It could be, or they take up more glucose when it's available. It could be that they're healthier, so they're able to carry out metabolism faster, so they're taking up more glucose. So more needs to be done on this, but again, it looks like it's a good result in the sense that the cells have more glucose, which again, they need to build their outer segment and also get more ATP. Okay, one more approach to getting the cones more fuel, and that was to induce the cones to use more lactate. Now, lactate is also available from an adjacent cell type called Mueller glia, which can carry out glycolysis and secrete lactate. So we thought maybe we could also get the cones to use that as their fuel source instead of relying on glucose. So Sawyer Shu joined the lab as a postdoc, and he really did an amazing amount of work. So over here are the cone counts when he introduced a number of different genes. Each of these bars is a different gene delivered by AAV, expressed only in cones with a cone-specific promoter. And here's the control. This is a mouse mutant, the RD1, that's very rapid. So that's the number of cones left in the control. Each bar, again, is the number of cones after we introduced one of these genes. And I know you can't read these, but these are genes that had to do with glucose metabolism or glucose transport or glycolysis. And we had only really one winner, which was called TIC-SNP. So what is TIC-SNP? So TIC-SNP is an alpha-arrestin protein, and these are proteins that bind to other proteins to carry out or change the function of the other proteins. So one of the things it's famous for is it binds to thioredoxin. In fact, that's how it got the name. It's a thioredoxin binding protein. Another function is to remove the glucose transporter from the surface. Many people who work on diabetes are interested in this. Another study we saw had TIC-SNP improving mitochondrial metabolism, perhaps by forcing the cells or allowing the cells to use these alternative fuels, not just glucose, but lactate, lipids, ketones, and amino acids. So we saw that TXNP benefited the cones. We didn't know how. And one of the things that Sawyer did is make many different mutations in TXNP to see if, first of all, could we make even a more active allele? And secondly, could it reveal to us what portion of TXNP was important for this rescue? So he made a lot of mutations. It turns out one of the first ones he made was the best. So subsequent, many mutations didn't improve anything. But what he found was that if he changed the interacting amino acid, a cysteine, that was necessary to interact with a thioredoxin, if he changed that to a serine so it could no longer interact, we had a more powerful rescue on the cones. We're just guessing. That's because thioredoxin is useful for fighting oxidative damage, and there is oxidative damage going on in these models. It also frees TXNP to go do other things, including these other things I have listed here. So this C247S allele of TXNP turned out to be our winner. Now, how does it work and how? Well, I'll show you the quantification first. Again, these are the cones stained in green, and we're looking at a control in the center here, which has about 6,600 cones. And here's the allele that I mentioned, the C247S. We have 13,000. And we tested this in 3 different mouse models. They all have improvement in the cone numbers as shown here. So the RD1, RD10, and knockout of rhodopsin. So some of you may know these strains. These are kind of standard strains that we use in the field for looking at retinitis pigmentosa. So it works across 3 different disease genes. So again, we're hoping for a gene-agnostic treatment. So this is good news for us. We also looked at the optomotor, and that's shown here. This is the RD10 optomotor. Red shows us where TXNP is present, and this is the rhodopsin knockout. So again, it's helping with vision in 2 different strains. And then we started looking at how might it work, and because we had read this paper about alternative fuels being important, we thought we would look at that. And there's a couple different enzymes that are needed for those alternative fuels to be used. So to use lactate, a cell uses a form of the lactate dehydrogenase gene, LDHB. Lactate then gets converted to pyruvate, and pyruvate can then enter into the TCA cycle eventually. Ketone catalysis uses OXCT1, and fatty acid catalysis uses the CPT1A. So we wanted to know if it's using any of these pathways, and for that, we introduced a knockdown construct along with TXNP to ask, would TXNP still rescue if we removed, for example, LDHB or either of these? And that's shown here. So again, LDHB was delivered, or all of them were delivered individually as shRNAs. We put in our cone GFP and TXNP and counted cones. And what we found was that that TXNP required LDHB to rescue. And I'll just draw your attention to these panels over here. So this is the control without any treatment or GFP. It looks about the same. This is a non-targeting shRNA. This is by itself, no TXNP, an shRNA to LDHB. It doesn't seem to affect much, which means that the cones are not normally doing much with lactate. Here's TXNP with a non-targeting SH. We got good rescue. And if we knock down LDHB at the same time we add TXNP, we lose almost all the rescue. So it looks like TXNP is somehow inducing the cells to use lactate to their benefit. We looked at the ketone and fatty acid proteins by, again, using shRNAs and did not affect the TXNP rescue. So something about TXNP gets them using the lactate. We don't know how. We looked at protein and RNA for LDHB, and it's the same as in the control. So it's not inducing transcription or translation. So we're not sure how this works, but at least we know now it gives the cells the ability to use their lactate. We also looked at mitochondria, and here's some data showing micrographs of controlled TXNP and TXNP 247S mitochondria size and shape. We measured them, and they were bigger if we had TXNP on board. We looked also at the health, looking at this dye called JC1. If the mitochondria are healthy, we get a pink aggregate. If they're not healthy, it's green. And you can introduce the dye. These are, again, all in vivo. Then immediately take it out and look at green versus pink. And what you can see is that we have more pink when we have TXNP on board. And these are the quantifications of the ratios. Again, suggesting these mitochondria are bigger and functioning better. We also did a functional test, I'll tell you down here, to look at the sodium-potassium ATP pump function, again with a dye. And when TXNP's on board, more pink means more active pump. relative to the control. So it looks like TXNP is benefiting mitochondria and requiring the cells to use their lactate. Now, one other protein partner that was in the literature for TXNP was the PARP protein. And for reasons I don't quite understand, so PARP is involved in DNA repair. And if you remove it, if you make a mouse knockout of PARP, the mitochondria are healthier. So we wondered, is TXNP interacting with PARP, somehow maybe taking it out of action, and that's part of the rescue activity of TXNP that leads to better mitochondria? And maybe just a PARP knockout by itself, no TXNP, might it also benefit cones? So we got the PARP knockout, we bred it to one of the RD mutations. We also just kept it as a separate series of alleles. And we asked if we just have the PARP knockout in the RD1 background, do the cells live longer? Do the cones live longer? The answer was no, but we did get improved mitochondria, and that's quantified here. So improved mitochondria without TXNP, just improved mitochondria, did not benefit the RD1 cones. If we included TXNP, same result. That is, the TXNP was doing its thing. It didn't matter if we removed PARP. It was the same with and without PARP, TXNP could rescue. So there is a benefit to mitochondria. We're not sure it has anything to do with PARP. And making just better mitochondria is not sufficient. We tried one more thing. We're thinking that we have to address more than one of these problems for the cones. So I told you there's metabolism shortages, there's oxidative damage, there's inflammation. We think we're going to have to address multiple problems to really get a long-lasting benefit to cones. So we've started to do combinations. This is one that we like. This is adding TXNP by itself. So you're looking at a flat mount of the retina stained with cone opsin. That's the photopigment protein. And what you see is the staining is kind of amorphous. It's more or less around the cells and a little bit in a stub here that used to be an outer segment. But if we put in the TXNP and we add this transcription factor NRF2 that I'll tell you about next, which fights oxidative damage, now we have a much more organized structure. It's reminiscent of outer segments. It's not really a good outer segment, but you can see there's organized structures here by putting these two things together. So again, we think the direction we have to go in is adding multiple genes to combat multiple problems. Okay, to summarize then TXNP, we think that TXNP is benefiting by affecting a fuel switch. So normally glucose is coming in through the glucose transporter. It then goes through glycolysis. Usually it ends up in lactate secretion. So TXNP removes the glucose transporter from the surface. So in the TXNP treatment, it can't get glucose. But instead, it seems to induce the cells, mechanism unclear, to use lactate through LDHB, which can create pyruvate, which can end up fueling the TCA cycle. In addition, there's a benefit to mitochondrial health. Now, that could be secondary to something that's going on here, having more pyruvate, having more building blocks, having more ATP, or it could be an independent function of TXNP, and we're not sure. There's no literature on this point. Okay, so I just told you about 3 treatments. One is gluconeogenesis, have the cones make their own glucose. Another is to give the RPE the ability to take up lactate, we presume from the blood, and thereby release the glucose to the cones. And also inducing the cones to, instead of being reliant on glucose, to now start using the lactate. So this is a potential path forward to help the cones survive. Okay, I want to tell you now about oxidative stress, which has been measured in tissue from patients as well as in these mice. So there's oxidized proteins, lipids, and nucleic acids. Now, there are ways to fight oxidative damage. The cells actually have quite a few enzymes and genes that are involved in antioxidation control. One of them is called NRF2, which I already mentioned. Another is PGC1 alpha. They drive hundreds of genes. Some of them are the same genes. So we introduced those as well as directly coded some antioxidation enzymes in the AAVs and delivered to these same mouse models. And we get a nice rescue. So again, here's a flat mount of a GFP-only treated diseased retina. Again, a lot of the central cones are gone. And if we put in one of these antioxidation treatments, we can get a better preservation of cones. And this is the quantification. So this is the age of the animal. This is the number of cones in the center. The green shows the GFP control. Cones are dying rapidly through this period. PGC1 alpha, for some reason that we don't understand, was deleterious to cones. SOD2 and catalase, which are enzymes that detox free radicals, was beneficial, but it wore off. But NRF2, this transcription factor, had a more perdurant kind of effect, and it was higher rescue. It also gave better optomotor, which is shown here in this, in this brown curve relative to green, which is the GFP only, or white, which is the no vector control. So acuity again is on the y-axis and age is on the x-axis. So NRF2 was really good in the RP models, in all 3 RP models. So we started thinking about it perhaps as being a good treatment for age-related macular degeneration. I'm sure many of you know about this. It's becoming more common as we age. Quite a few people have it. It costs quite a bit. We don't have a treatment for the most prevalent form. 90% of patients with AMD have something called the dry form. The wet form is leaky blood vessels, and that can be treated with injections of anti-VEGF, but that doesn't help 90% of the people. Doesn't even help all of the people with wet AMD. So we thought that perhaps we could start to look at some of these vectors if we had good animal models for AMD. It turns out we don't. So age-related macular degeneration has the word macula in it because that's a central part of our eye where our high acuity vision takes place. And for reasons that are unknown, that's the most sensitive place in terms of aging and some genetic hits. So there's some GWAS hits for age-related macular degeneration. There's some risk factors like smoking and obesity. We don't really understand the etiology here. And the animals that we use don't have a macula. Again, that's a specialized area right in the middle of the retina where we have our high acuity vision. None of the animals that we typically work with have a macula. So we can't model it very well, but we thought, well, at least if we could look at the effects of our vector on different cell types in different acute models, which mimic some aspects of AMD, maybe we can see if we have a potential AMD therapeutic. So what we did is we looked again at our retinitis pigmentosa models at the RPE. So what's shown up here in the top is the apical surface, the surface facing the photoreceptors of a wild type animal, and each of these little hexagons is an RPE cell. They have many microvilli shown over here on the right. These little microvilli surround the photoreceptors. This is where nutrient exchange takes place and waste removal takes place between the RPE and the photoreceptors. If we look at one of the mutants for retinitis pigmentosa, we're losing RPE and we're losing all of the apical microvilli. So these RPE cells are degenerating quite rapidly in this model. And again, they don't express the disease gene, so it's secondary causes that are leading to the RPE cell death. So we wondered again if it might be oxidative damage or other things that are being experienced by the cones. And so we put in this NRF2 transcription factor driven by an RPE specific promoter. And you can see that we can rescue or preserve the RPE. It's got beautiful apical microvilli, and presumably it's providing better function now for the cones. So given this and the fact that in age-related macular degeneration, the RPE cells are sick and the cones are sick, we thought, well, maybe since it's capable of rescuing these RPE cells and the photoreceptors in retinitis pigmentosa, maybe it can do the same in another model. And the model that most people use is called sodium iodate damage. So sodium iodate is an oxidizing chemical. You can inject it IP or through tail vein, and it seems like it preferentially kills off the RPE, even in a few days. You can also have cone death and choroid leakage, the blood vessel leakage. So we thought we would try NRF2 in the sodium iodate model to see if it would prevent damage there. So we do the same thing as I've been telling you. We inject our AAVs early. We let the animal grow up. The AAVs are expressing now. We then look at in-life assays. The ERG is an electroretinogram, and it tells us the overall electric activity in the eye. OCT is a very nice imaging modality where you can look in the eye and see all the layers of the retina. So we do those at a few time points. And then we inject the sodium iodate, IP, and it kills the RPE. We take out the eye, again, do the same assays I already told you about. And this time we're also staining the RPE and quantifying it with an antibody or a marker called phalloidin. And so what we find is that if we infect with the best one, NRF2, and again, this is directed for expression only in the RPE, we prevent the loss of the RPE cells. So hope you can see these nice hexagons. So in a control saline injection, no sodium iodate, if we have NRF2 or a control GFP vector, which you see with the green, hopefully you can see there's these nice red hexagons. If sodium iodate is injected over here, without the NRF2, just a GFP, we lose almost every single RPE cell. But if we have NRF2 on board, we get these beautiful hexagons again, and that's quantified over here. We basically lose all the RPE cells from sodium iodate with a control virus, but we save them with the Nerf2. So it's a very good, I mean, this is a very acute damage, and I was worried it would not be that protective, but somehow it protects really well. We also counted cones, and these are flat mounts stained with cone arrestin. Again, if we have a saline, not sodium iodate injected, the control and the Nerf2 infected look like they have very nice cones. If we put in the sodium iodate without Nerf2, most of the cones die, or they shrink their outer segments and then die. And if we have the Nerf2, we preserve not all of them, but most of them. And if we look at cross sections down here, again, saline, we get these are very organized laminated retinas with a thick outer nuclear layer, which is where mostly rods reside, cones as well, but 95% of the cells in this layer are rods. And again, the control, no sodium iodate, lots of rods. When we put in the sodium iodate, we preserve the outer nuclear layer, and this is all quantified over here, but you can see the outer nuclear layer is really destroyed by the control virus plus sodium iodate. So the NRF2 is then preventing cone and rod death. We were also wondering whether there was non-autonomous effects, and so we looked at a partial infection. We also wanted to know would it rescue in adults, because again, people would be treated as adults, not as neonates. And so we infected and got a partial infection, and we could track where the infection was because we see GFP, where we have Nerf2. So these little boxes represent different places where we have no infection or almost no infection in these top two. And you can see very aberrant morphologies for the RPE. But if we have a lot of Nerf2 in the area, shown by the green spots, we preserve the RPE. And this is a partial where here's an area with no or little GFP, here's more, and you can see the transition from protected to susceptible. So it looks like we can rescue if we inject in the adult. And whatever's going on by the Nerf2 delivery, it keeps the effect local. Okay, so just to summarize, Nerf2 therapy, we looked at RNAs, we looked at markers of oxidative damage, and it does provide for antioxidation. I didn't mention, but it's kind of a twofer. It also reduces inflammation. It's negatively transcriptionally regulates IL-6 and IL-1 beta, which are very high up in the hierarchy of the immune system. There are a lot of RNAs we found that had to do with metabolism, and we're not sure how to interpret those. We can express it only in the RPE, which is what I just showed you, the best one promoter, and we're thinking that might be our first approach to AMD because it's very safe. We keep the transcription factor very local to the RPE, which dies in AMD. We can also express it in cones and the RPE, which is what I showed you for the retinitis pigmentosa models, and it protects again in the RPE and the cones, and it can protect from the sodium iodate damage. So this work was started by Wenjin Zhang and carried on by David Wu, and now Apollonia Gardner is carrying on more of the work with sodium iodate with a team of people in my lab. And we're also really fortunate to work with Spark Therapeutics on this with the team of people listed here. So we're hoping that the best one Nerf2 would go forward in the clinic for AMD. Okay, last thing I'll tell you about is inflammation. So again, most diseases have an inflammatory process going on. So Sean Wang, who was a medical student at the time, was interested in whether inflammation was negative or positive. Sometimes inflammation can be positive. So microglia are the macrophages in the CNS, and they have a number of roles in development, in disease, maybe in other processes as well. You can see depicted here a few of the things that they're capable of. And Sean wanted to know, is there a role for inflammation and perhaps a role for microglia in degeneration? So if you look at a wild type mouse section, which is shown here, here's this outer nuclear layer that I told you is where the rods and cones reside. Green shows us where the microglia reside. And in a normal wild type animal, they're not in the outer nuclear layer. They're in these other layers. But as degeneration proceeds, this outer nuclear layer shrinks. All the rods die, and we're left with one layer of cones, which you can't see very well here. But these green spots are now representing microglia that have moved into the outer nuclear layer that's left. That is, they're surrounding the cones. And there's also an upregulation of a variety of inflammatory RNAs. So we quantified the GFP cells in the outer nuclear layer in wild type and disease, and there's quite a few that go in there. Here's just some of the inflammatory cytokines that go up also. They're increased over age during cone degeneration. So one experiment we did then was to wipe out the microglia with a drug, a PLX 5566 drug, and we confirmed that the microglia were gone. And we thought, well, if the microglia are in the outer nuclear layer, secreting these cytokines, harming the cones and or eating the cones, if we remove the microglia, the cones will live longer. Turned out that was incorrect. We got no benefit or harm to the cones by removing the microglia. So Sean then thought, well, let's just dampen down inflammation. There's other ways, there's other molecules, cells that might be involved. And so he tested a number of molecules, again using AAV, to ask, could we benefit cones by, for example, having fractalkine secreted? Fractalkine can be secreted or be membrane bound. CD47, some of you may know, it's kind of famous now. It's on the surface of tumor cells, and it tells the macrophages, don't eat me. And it has a couple receptors, SERP alpha and thrombospondin. TGF beta 1, you may also know, can be secreted, and it works through a particular set of receptors. So Sean manipulated all of these, as well as CD200 and TREM2, and what he found was secreted fractalkine, but not membrane bound, would rescue. CD47 on the surface of cones protected the cones, and we think it's through SERP alpha. And TGF beta 1 secreted from cones or by cones also protects cones. Now, we're not sure what the mechanism is for these rescues, and we're interested in that. How does TGF beta 1, for example, produce this survival effect? We know it requires microglia, because if we take away the microglia, TGF beta 1 no longer rescues. We're not sure about fractalkine. If you remove the known fractalkine receptor, it still rescues. So kind of interesting there. Okay, so just to summarize a lot of work that Sean did over many, many years in 3 papers. So the microglia reside in the photoreceptor layer throughout the period of cone degeneration, but they don't cause the cone degeneration. It's just a correlation. We can put in secreted fractalkine. It's actually secreted from the RPE, or TGF beta 1 secreted from cones, or CD47 on the surface of cones, and all of them improve cone survival, again, across all the RP models. They also improve cone vision as assayed by optomotor. And we'd like to understand how these work. Still working on that. Okay, I do have, just give you 2 more minutes and then I'm done. One more thing, which was a project carried out by Ryoji Amamoto, who was a postdoc in the lab. And we were very intrigued by a finding over many years of doing this work that cones in the dorsal periphery seemed very long-lasting in all the degeneration models we studied, much longer than one would predict from the rate of degeneration everywhere else in the retina. So this is a flat mount of a wild-type animal stained with a cone marker. This is the RD1 mouse mutant I've been telling you about. Most of the central cones are gone by postnatal day 40. But if we look even later, at 1 year of age, in the RD1, RD10, or row null, these are all the models we use, the dorsal cones, dorsalmost cones, seem to live longer. And there seems to be a fairly sharp line demarcating the long-lived cones and the rest of the retina. These are all quantified here. We wondered why. We did RNA-seq to find if there were some genes enriched in the periphery relative to the center, and there was one winner called LDH1A1, and we showed that it was expressed in Mueller glia in the dorsal periphery. So then we wanted to know, where's the signaling? So that enzyme makes retinoic acid. Retinoic acid can come out of cells, can enter cells, bind to a receptor called RAR alpha or others, but RAR alpha with RA bound can turn on transcription of genes that have a certain response element. And we could get a reporter mouse and ask, where is the RA signaling in these mice? Turns out it's exactly where the cones live longer. So that's what's shown here. This beta gal is the readout of where we have active RA signaling, and it's just where we have the prolonged survival of cones. So that said to us, well, maybe this ALDH1A1 enzyme is actually producing RA. The Mueller glia are the ones who are producing it. The signaling we saw was in the Mueller glia, and then we wanted to know, is that really what's protecting the cones? So we got a lot of knockouts from our colleagues. This was a 10 allele mouse where we could turn on Cre, which knocked out all of the enzymes that can make retinoic acid. And if we have Cre activity, which is shown here, versus no Cre activity in the same genetic background, the cone number goes down, which means we do need the production of RA to get the prolonged cone survival. Moreover, if we put in a retinoic acid receptor that's constitutively active transcriptionally, we preserve cones. So that's shown here. If we put in the RAR alpha relative to a fluorescent protein control, we get greater survival of the cones. So it's necessary and sufficient. We also looked at human tissue, and this is now center and peripheral RNA levels for this enzyme. These are 5 different individuals. In the periphery, we have more of this retinoic acid synthetic enzyme. So it's possible it's also functional in the human. So just to summarize then, normally the retinoic acid is being produced by Mueller glia in the dorsal periphery. It is necessary because if we take away the synthetic capacity, we lose not all of the cones, but many of the cones. And if we add it to a central area that doesn't normally have it, we can preserve the cones, so it's sufficient. Okay, to summarize then, these efforts to get longer cone survival and function benefited from several types of treatments. This augmentation of nutrition, which I showed you, was all focused around getting them more glucose or more carbon source. We can upregulate the antioxidation pathways with NRF2 and get better survival, both of photoreceptors and the RPE. We think that we should be doing combinatorics, and again, I showed you one example of putting in the gluconeogenesis genes with NRF2, and we got an additive amount of survival. We can manipulate inflammation and get better survival, and retinoic acid signaling in Mueller glia seems to benefit the cones. So what are we doing now? We would like to test in large animal models. There are some large animal models for retinitis pigmentosa, not for AMD, unfortunately. We want to know again about infecting later. I showed you one example. There's actually 2 examples now where we know we can infect later and still affect a rescue. Combinations, we still have to keep going on combinations. And as I mentioned, the first perhaps clinical treatment will be the best one NRF2 vector, which is restricted to the RPE for AMD. And we've been really fortunate to have a number of very good funding sources, and we're collaborating, as I mentioned, with Spark to try to take some of these forward to the clinic. So I'll stop there and be glad to take questions.
Before we go for asking questions, I request Dean Rosenblatt to present a token of appreciation to Dr. Cepko.
Something to remember us by, but thank you so much for an outstanding lecture, and thank you so much for being here with us.
Okay, we have few minutes for a few questions. Who wants to go first? Go ahead.
So I was curious about the large animal models. You mentioned that some of your models don't have a macula. Are you looking at transgenic marmosets?
No, unfortunately, we don't have access to, I don't know of any models that have been made. I mean, they do have their own foveas, though. They have their own macula. So you don't have to make the transgenic to get a macula. So you could treat them. Now, you probably know that it's a disease of aging, and they're very expensive, and it's hard to get them these days. So we're not planning that, but I keep asking other people if they would inject for us. But so far, no.
Thank you for an excellent presentation. I was wondering, you mentioned a couple of times that sometimes these treatments wear off.
Yeah.
But these are all post-mitotic cells. I'm wondering what do you think is going on?
So I think, so he was asking whether these treatments wear off and why. We think it's because so far we haven't stopped all of the problems. So if there's oxidative damage, metabolic shortcomings, inflammation, and we're treating just one of those, the other processes are still going on. And so we think we're getting a benefit from knocking down one of those pathways. But until we arrest all of those pathways, I don't think we're going to have really long-lasting benefits.
Thanks. That was a really lovely talk. Super clear. I was wondering about the last part that you talked about the cone's survival in the periphery, and whether you have any idea of why that metabolism or process, the retinoic acid, is being made there in the first place, and if you know whether that happens in primates in any particular area of the retina.
So that's a great question. So I can tell you a little bit. So we're looking also at the patterning of the retina early in development. So most of the work's done in chick, but some of it we've extended to humans. And what we see is that in the dorsal regions, in the early stages of development, there's retinoic acid synthetic capacity. So it's there, and we've manipulated the retinoic acid in the chick, and we find that it's required for setting up the high acuity area in the chicken. So there's early patterning, which involves RA signaling very early before all the neurons are born. So that early patterning event requires retinoic acid. It's dorsal, and it's small domain ventral. It's also in the human with the same expression pattern. So one possibility is it's there for an early patterning role and it just doesn't get turned off. So it didn't evolve to protect dorsal cones, but it just didn't get turned off. It wasn't deleterious. So that's one possibility. Another is that, yes, it was there for patterning purposes, but then it was retained there for the function in later life to preserve those cones. And why those cones, I don't know. Haven't been able to come up with a good reason for those, except to say it was there for it was just there for the patterning role. So it could be used there for persistence of cones. That's one possibility as well. But really, we don't know.
One last question.
I guess the question is, fine. So my question is more regarding for you, targeting inflammatory response. Like for instance, you showed microglia and you showed that they do like migrate to the site where you have the cones dying out. I take it this is like due to damp signaling. Did you check by any chance if they were in a phenotypic like M0, M1, or M2? Because to my understanding, the M2 would be their activated state, and when you depleted them, there was not, it was not exactly beneficial. But I know that from M1 it can go to M2, and it's more like it has a more protective function. Also, to my understanding, there's also like new treatments. For instance, since microglia are very active in neurodegenerative diseases, there's treatments that do target inflammation, like using beta-caryophyllin that can target, I think it's a CB2 receptor, and then make it shift from M1 to M2.
Yeah, those are great questions, and we really haven't delved into that, and we should. So I really can't answer what stage they're in or state they're in. When we found that they weren't, getting rid of them was not beneficial, we kind of stopped. We lost a little bit of interest in microglia. So we got rid of all those inflammatory cytokine RNAs when we wiped out the microglia. So they're required to make those, but that didn't benefit the cones. The cells themselves, when they were gone, there was no benefit to the cones. So we didn't really pursue microglia as much as we would have if we thought that they were actually engulfing the cones or those cytokine RNAs were deleterious. So that's why we kind of moved on.
I invite all of you to join us for the reception in the faculty alumni lounge where you can continue the conversation. Dr. Cepko, thank you so much for this.